Hello, this is a marine ecology lecture about continental shelf ecosystems. I'm picking for the background a simple diagram of continental shelves to remind you that continental shelves are the extension of the continental land masses into the oceans. So it's basically just the water-covered margins of the continents of the world, which are very shallow waters in comparison with the ocean basins, which are geologically different under a thinner and more low-lying type of Earth's crust called the oceanic crust. So the continental shelf is this sort of generally flat-sloping extension of the continent near the coast, and then at the edge of the continental shelf it slopes off, which we call the continental slope, until it eventually meets the abyssal plain of the deep sea floor. The continental shelves of the world cover about 8% of the ocean floor. So most of the ocean is deeper waters away from the continents, not continental shelves. And so continental shelves are just kind of this marginal area near the shores. And there are a few places in the world where there are shallow waters that are not near a large continental landmass, like off of New Zealand. But for the most part, the areas that we would consider to be continental shelves are around the large terrestrial landmasses that we call continents. Some places have wider continental shelves than others. In some places, just a short distance off the coast, you'll drop off into deep water. But where I'm giving this lecture from, the Gulf coast of Florida, the Gulf of Mexico coast of Florida, the continental shelf is very broad, and one would have to travel actually hundreds of kilometers over flatly sloping shallow bottom until they reach the shelf edge in the west coast of Florida. So to put continental shelf habitats in context, they are a much smaller component of the world's oceans than sort of the open ocean and deep sea, both in terms of area and volume. So they're only about 8% of the sea would be considered continental shelf waters. But in that 8%, there's high productivity, and a lot of the ocean's diversity is found in shallow water continental shelf ecosystems with interesting structural habitats like reefs. And there are some really specialized habitats within continental shelves like estuaries, reefs, mangroves, salt marshes, and seagrass beds. And those are an even smaller component of the surface area of the Earth's oceans than the continental shelves at large, but they have an extremely high diversity and productivity. So continental shelves are sort of this intermediate level habitat where they're not sort of as vast and barren as the pelagic and deep sea, but they're not as dense and overflowing with life as these really specialized habitats like estuaries, reefs, mangroves, salt marsh, and seagrass, which are sort of like subcomponents of the continental shelf ecosystems of the world. So to give an example of continental shelf productivity and its relationship to human life and ocean ecosystems at large, I want to talk about this particular area of the world's continental shelves in the continental shelves off the coast of the U.S. state of Alaska. So, and that's this area here. So these waters are very productive due to current patterns that cause a lot of upwelling of nutrients. And that upwelling of nutrients supports phytoplankton productivity, which in turn supports zooplankton productivity, which in turn supports small fishes, large fishes, seabirds, marine mammals, and even humans. There are many important fisheries in the waters off of Alaska, everything from benthic crustaceans like snow crabs to demersal fishes like the Alaska pollock. This beautiful fish here is a member of the cod family. One of the distinguishing features of some members of the cod family is that they have not one, not two, but three dorsal fins. And oftentimes they'll have a little whisker, although I'm not sure if the pollock has that or not. So this is one of the most abundant predatory fishes in the continental shelf region of Alaska, and there's a large trawl fishery for it. So a trawl fishery is a type of fishery where there's a big boat that tows a big net behind it, and that net is called a trawl. So all of the unfortunate Alaska pollock get caught in the trawl, dragged on board the ship and processed, and eventually they make their way into the maybe most famous fish sandwich in the world, the McDonald's fish fillet sandwiches made from the fillets of this beauty of the sea. So how is it that continental shelves are so productive and provide us with this bounty of fish fillet sandwiches? It's complicated. So to understand the productivity and the diversity, which means the variety of species of continental shelves, we need to think about the nutrient sources to continental shelves that fuel the primary productivity, the waves and current patterns on continental shelves and how they're different from open sea areas, the light and depth situation on continental shelves, and the substrate characteristics. So the substrate is the bottom, and it turns out that the composition of the bottom is very important for determining what type of life and what diversity of life is found in continental shelves. So we'll go through it in this lecture and talk about each of these things in some more detail. So let's talk about nutrient sources first. We know from the primary production lectures that nutrients are incredibly important to primary productivity. So where the nutrients are coming from is a major question that ecologists need to ask and answer to understand the productivity of different environments. And so on continental shelves, there's two major kinds of nutrient sources, two major classes of nutrient sources that have to be considered. There are the allochthonous nutrient sources and the autochthonous nutrient sources. So allo means from outside or other. So allochthonous means nutrient sources that are not from the continental shelf, that they're nutrients being delivered to the continental shelves from other environments, and those environments include the deep sea, when you have upwelling of deep sea water onto the continental shelf, and they include the continental land masses as rivers and runoff carry nutrients from the land into the continental shelf waters. So those are two outside or allochthonous sources of nutrients to the continental shelves. And then autochthonous means sort of like self-generated or regenerated. So that's sort of like the nutrients that are already bound up in some form on the continental shelf and just keep recirculating throughout the continental shelf ecosystem. This diagram here visually explains what I was just talking about with the two allochthonous nutrient sources, like the runoff from the land and the upwelling from the deep sea. Those are both sources from off the continental shelf. And then the autochthonous nutrient source, which was the sort of recycling of nutrients within the continental shelf environment. So the autochthonous recycling of nutrients within the continental shelf environment is strongly affected by waves and currents. So waves and currents have a major role in sort of recycling the nutrients within the continental shelf environment because they can enhance something called nutrient flux. Flux is a fancy science-sounding term, and it's important that we define it carefully. So if you look it up in the dictionary, the definition for flux that we want is definition number 6, the rate of transfer of fluid particles or energy across a given surface. So that given surface could be the sea bottom, it could be the boundary between the surface ocean and the deep ocean, it could be the boundary between the outside of an organism and the inside of an organism, if it's the flux into or out of a cell, for example. So you could be thinking of the flux at the small scale of a single cell or the large scale of the sea bottom or different layers in the water column. So the transfer of fluid particles or energy across a given surface. Remember, a surface is like a two-dimensional plane, and so you're sort of quantifying how much is crossing that plane, either into or out of the area that you're considering. So waves and currents enhance flux. And the two types of flux that waves and currents enhance are, one, flux from the bottom to the water column, and two, flux from the water column to the organisms in the water. And both of these fluxes can go the other way as well, so from the water column to the bottom or from the organisms to the water columm. So this types of flux is actually a two-way thing. But I'll go through so you can kind of imagine how the flux from the bottom to the water column might work. So first, organic waste settles to the bottom. It could be in the form of sinking fish or fecal pellets or some kind of particles of organic matter sink to the bottom. And then they decompose in the bottom, releasing nutrients into the sediment pore waters. So pore water means the water that's in the interstitial spaces between the sediment. So it's sort of like the water that's within the sand or mud. So high nutrient levels like nitrogen and phosphorus build up in the mud and sand beneath the seafloor. And then when there's waves or currents, that can stir up those nutrients and reintroduce them into the water, sort of increasing the flux of nutrients out of the bottom and into the water. Then there can be a flux from the water column to organisms. So organisms like, let's say, here's a phytoplankton cell. Let's say it's a diatom. It's got a nucleus and some other things in the cell. This diatom needs to get nutrients into itself. And if there is turbulent water motion, that can help deliver the nutrients across this sort of diffusional layer into the diatom cell. The same thing if this, instead of being a single-celled alga, it was a seaweed or something, that could help deliver the dissolved materials more efficiently across that boundary layer, sort of the area around the cell, into the cell. Water motion can also help heterotrophic organisms like a sea anemone. So sea anemone is relatively easy to draw. It's just sort of a little trapezoid with tentacles on the top. So water motion can sort of help deliver particles of food into the tentacles of a sessile organism, increasing sort of the flux of food particles from the water column into the feeding apparatus of this organism. So on a continental shelf, water motion that is in the form of surface currents or waves is more likely to interact with the bottom simply because the water is shallower. So when a wave passes by in the water above, there is motion in the water down to a certain depth, not all the way to the bottom if the bottom is deep, but if the bottom is shallow, it will be within this zone of wave-influenced currents. And that depth of wave influence is proportional to the wavelength, so proportional to the distance between successive troughs. It's actually about 1/2 of that distance, 1/2 of the wavelength down is sort of the level, the minimum level, the deepest level in the water that the wave motion would be felt. And so if the bottom is shallower than that, it means that the waves are rubbing against the bottom. When the waves rub against the bottom and sort of rock back and forth, you can see it when you look at the bottom, because you'll see all of these ripples in the sand that form from that lateral current washing back and forth over them as the waves pass. So these currents from waves and other sources of currents like tides, they can transport and resuspend sediment, so they might erode the sediment away, or they might bring more sediment in and deposit it so they can move the sediments around, and they can also mix nutrients up from the bottom into the water column, which has important consequences for primary producers, which now have access to those nutrients. On the topic of waves and their influence on the bottom environment or not, I think this is a good time to bring up one of the interesting things about the west coast of Florida in comparison with the east coast of Florida. So the east coast of Florida is, at least the northeast coast of Florida, is known for having pretty nice waves. There's a famous surfer named Kelly Slater who learned to surf and got good at surfing on the east coast of Florida. But the west coast of Florida, you go to the beach there, and as often as not, it just looks like a lake. So why is it? Why would the waves be so small on the west coast of Florida in comparison to the relatively large waves from the east coast of Florida? And it's not just a wind thing, because there can be plenty of days where there's relatively strong wind on the west coast of Florida, and this is the wind from a wind report from a surf report. And yet, even when there's this relatively strong wind, you often still have flat surf with no waves. So why is this? What gives? Well, it turns out that the direction of the wind is very important. So at latitudes below about 30 degrees north, so sort of from the equator to 30 degrees north, the prevailing winds are from the east. So that means that waves are being pushed by those winds against the east coast of Florida, but waves are moving away from the west coast of Florida, and so the wind is sort of just touching the water when it gets here, and it hasn't had time to build up any waves, and any waves that it does build up, it sends to the opposite side where they impact the other side of the Gulf of Mexico. So the prevailing wind direction is one of the main reasons, if not the main reason, that the waves are small on the west coast of Florida. Another reason is fetch. So fetch is the sort of distance that waves have to build up. So in the Atlantic Ocean, we can't even see the entire Atlantic Ocean on this diagram, but it's really wide. And so the winds have a long stretch of ocean across which to push up waves that get bigger and bigger and bigger as they sort of build momentum and get pushed and pushed by the wind as they're going across. In the Gulf of Mexico, even if the wind is blowing the right direction across the Gulf of Mexico, which it does sometimes in the wintertime, it doesn't have as long to build up big waves before they run out of space. This lack of fetch is the same reason that waves generally don't get very large in small lakes, because even if the wind is blowing strong, the waves only have a kilometer or two to build up before they smash into the opposite shore of the lake, and they're done. And then water depth is a third reason. Because the continental shelf is so wide on the west coast of Florida, the waves begin to feel the friction of the shallow water, and that sort of affects their movement and size. And it doesn't completely eliminate them, but it sort of takes away some of the energy from the waves as they approach the coast of the west coast of Florida. So the combination of these three factors is bad news for surfers on the west coast of Florida. It's why we rarely have big waves on the west coast of Florida. What does this have to do with marine ecology? Well, it means that the sea bottom environment on the west coast of Florida is much less disturbed by waves. Finer sediments can accumulate because they're not resuspended, and this might affect the types of life that can live on the bottom on the west coast of Florida versus the east coast of Florida, where it's constantly disturbed by waves. So some types of life might do well in an environment where there's strong currents and waves. Other types of life might do better in an environment that's quieter, and that's one of the reasons that we see differences in the type of life on either side of the peninsula of Florida. Let's talk about some currents that are driven by tides and waves. Tides and winds. These are a little bit different than just the currents caused by wave influence, although wind-driven currents and waves relate to each other because waves are sort of the basis for wind-driven currents. Anyways, what you see in the diagram on the right here is strong currents rushing in through a narrow pass into a bay, and you can see that the currents are really swirling around this buoy, and you could imagine that organisms that are living on the bottom of this channel would be getting swept pretty strongly by these strong currents, for better or worse. And this picture taken underwater in the Florida Keys shows a type of soft coral called sea rod. And you can kind of see that as the current, which I think in this case is caused by waves at the surface, is sort of whooshing the stalks of this colonial sessile organism over, and they're sort of bending with the currents. So I have a little question here. How might currents help or hurt this organism? So currents can, of course, be good or bad. Some of it depends on the magnitude of the current and what type of organism the current is affecting. Well, a gentle current could help the sea rod by delivering particles of food into the many tentacles of the polyps on the sea rod. So sort of the more current passes by this sea rod in a day, the more particles are brought through its tentacles and the more food it gets. Also, the currents can help with the exchange of gases, like delivering oxygen and whisking away carbon dioxide from the respiration of the coral polyps. And there might also be nutrients like nitrogen and phosphorus that are delivered to the coral. So the coral can't eat nutrients directly, but there are symbiotic algae in the coral that use those inorganic forms of nutrients, and so having those nutrients brought in by the current could help the symbiotic algae that are in the coral grow. So there's actually quite a few ways that the currents could benefit this soft coral, and in general, we see that coral reefs grow most rich and lushly in areas where there is some current energy, either from waves or tidal currents or other wind-driven currents that deliver a steady flow of water that helps the organisms in all these ways. So some current can really be a good thing. Really strong current could be bad, though, because, I mean, if you can imagine a giant hurricane wave coming through and ripping this coral off at the stalk and killing it. So extreme waves and currents can be bad, but moderate waves and currents can increase the productivity of life. Currents really vary a lot in the continental margins, where the currents interact with constrictions in the seafloor. So you can see where currents get channeled as they have to go through narrow areas like this narrow opening here. The currents would be kind of like slow on average when they're far out here, but the currents would be accelerated stronger and stronger as they move closer and closer to the constriction. So the areas in these so-called bottlenecks, because they are narrow like the neck of a wine bottle, tend to have really strong currents and will have different organisms in them than other areas. Another thing that you can see is there's this sort of like delta of sediments that are deposited in these areas outside of the inlet here. And so it's sort of restructuring the seafloor as the strong currents, where they start to slow down again, are depositing the sediments that they'd carried and forming these sort of fan-like deposits of sediment that we would call tidal deltas. The current reverses direction in the Florida Keys. This body of water on the north side of the Florida Keys is called Florida Bay, and which is part of the Gulf of Mexico. And then this body of water, well, it's not called Florida. This is just called the Atlantic Ocean on the south side of the Florida Keys. And so when the tide is coming in, it runs this way through the channels, and when the tide is ebbing or going out, it runs this way. And so the deposits of sediment that form on this side are called the ebb tidal deltas, and the deposits that form as the tide is coming in are called the flood tidal deltas. In the Florida Keys, the water flow has been changed a lot by the construction of what they call causeways. So There are many places where you can kind of see it here, where in order to save money on building a bridge, they sort of built up the land between the islands, and so they would just have to build a short bridge between two piles of um, land that they built. And the net result is that the water exchange between Florida Bay and the Atlantic Ocean is less than it was before they constructed this highway with all of these filler islands and causeways built in there. And so that has changed the health of the ecosystem in some ways that aren't so good. Okay, why waves and currents matter is a complicated question, but I want to give you some kind of specific examples of how waves and currents might affect life living on the bottom. So here we've got some oysters. You can tell that they are in murky water, and oysters are sessile epibenthic organisms. They are non-moving organisms, and they like to live on the surface of the bottom. They don't want to be underneath the mud. They need to be on the surface, and they filter feed on particles that are brought in by the currents. So some currents are good. They'll deliver particles of food to the oysters, and they'll sort of whisk away fine sediments, muds that might bury the oysters. But it's a dicey thing. If it's possible for the currents to bring in a bunch of sediments and bury over the oysters, in which case the oysters will die. And it's also possible that maybe strong repeated waves could wash away the oysters, tumble them around, and carry them into some habitat that's not suitable for them. So getting buried by a pile of sediment brought in by the currents, or getting sort of exposed and blown away as the currents erode away the sediments around you, both of these things are problems that sea bottom organisms experience living in an area with strong currents. So many of those organisms deal with this problem of either getting buried or getting exposed by digging all the time. Oysters can't do that, so they're sort of more at the mercy of whether of what the sediment conditions are. But something like a worm, if too much sediment piled up on top of it, it could just crawl up closer to the surface. Whereas if the sediment eroded away around it, it could just crawl deeper down beneath the sediment. Another problem with waves and currents moving the sediment around for organisms is if there's a lot of sediment, like mineral grains mixed in with the edible pieces. So it's not just like plankton that's being brought in by the currents, but it's lots of little bits of rock that are being brought in by the currents. Oysters can't eat rock and mud. So if the water's full of more inedible particles than edible particles, they have a very hard time feeding, because their way of feeding is sort of filtering particles out of the water, and they have some abilities on the microscopic scale in their gills to pick out the edible particles from the inedible particles, but it's difficult and it takes a lot of energy. What they do is basically they wrap up the inedible particles, like the tiny microscopic bits of rock, in mucus, and that mucus wrapped mud becomes something that they call pseudofeces, which they then excrete from their shells. So they drop out these things that look like poop, but it's actually inedible material that they're clearing off of their gills, and so they would be dropping lots of these pseudofeces out onto the bottom. The thing is, it's an energy-intensive process, and if you're not getting very much food compared to the amount of inedible material that you're getting, then you're not going to meet your energetic needs, and you could get, no pun intended, pooped out. Okay, some organisms are really good at living in super high energy environments where there's tons of sediment moving around all the time. And those organisms have to have the ability to bury and unbury themselves rapidly. So they're mobile organisms that are good diggers. These crustaceans, decapod crustaceans here, called mole crabs, are very good diggers. They have sort of bullet-shaped bodies, and they're able to dig themselves into the sand backwards. If you pull them out of the sand, and they live on the shoreline areas where waves are washing back and forth and there's constant strong currents, they'll try to dig in to your hand, and it's very cute to feel their little legs wiggling as you're holding them in your hand. They filter feed with their antennae. So you can see the antennae are these feathery structures on this one here, and it'll be totally buried in the sand up to its eyeballs and its antennae, and just the antennae will be sticking out, and as the waves wash back and forth over the crab buried in the sand, it catches food particles in its feathery antennae. And that's what the video shows. So currents vary in relationship to the structure on the bottom, and the main reason is because there's friction between the water and the solid structures on the bottom. So like it's complicated though, because while there's the general trend that the closer you get to the bottom, like if this is the bottom here and this is the current, the closer you get to the bottom, generally the weaker the current will be due to friction with the bottom. And if you're scuba diving, you're having a hard time swimming against the current. So here's a scuba diver having a hard time swimming against the current. If that scuba diver was down at the bottom, that scuba diver would be able to swim against the current more easily because the current would be weaker. And if it was really bad, the diver could even sort of grab onto the bottom and pull themselves along on the bottom. So this is a strategy that you might have to use sometime if you're scuba diving and you get caught in strong currents. And so that's what's happening in the picture here, where someone's scuba diving in strong currents. But one of the complications to this whole thing about currents is that we've already talked about how where there is a bottleneck in the water, how currents that are sort of slow can get accelerated as they go through that bottleneck, and then they would slow down again outside the bottleneck. That effect can happen even if it's not squeeze from both sides. So like imagine this is the surface of the water and the bottom mounds up in an area. If the currents are sort of going along at a slow speed here, when the currents have to get over this mound, they have to increase in speed to sort of squeeze a bunch of water through that narrow area, and then they decrease in speed again when they get over deeper water again. So bumps and benthic structures accelerate the current faster. And this aspect affects life that lives on the bottom, particularly filter feeding organisms. So this thing here is an oyster reef mound. So it's a big pile of oysters that are glued to, they cement to each other and create this reef-like structure. It really is truly a reef made of oysters. And you can see that there's this sort of relatively slow currents that only go up to 6 centimeters per second away from the oyster reef. But over the oyster reef, because of the way it sticks up, the currents get up to 12 centimeters per second. And this is with a tall reef. But with a reef that was not so tall, it wouldn't interact with the currents very much, and you would still have like very slow currents over the reef. And so that's the kind of situation that they're talking about here with this low reef. So there's this sort of positive feedback effect where the taller an oyster reef gets, the faster it grows, because the taller oyster reef causes the currents to accelerate, and the currents accelerating means that more food gets delivered to the oysters at the top of the reef, and so the top of the reef grows even faster and it gets even taller until sometimes it'll actually breach the surface. This knowledge has been used by people that have been trying to restore reefs. We used to think that to restore a reef, you would want to, if you had like a pile of shells, you would want to spread them over as large an area as possible. But we had very little success with that sort of thin, low relief type of restoration. We found out it actually works better if we put all the oysters together in a mound instead of spreading them out, because that means that they get that acceleration effect that helps the oysters to grow and the reef to sustain itself. So this is an example of ecological knowledge applied to restoring habitats more effectively. So zooming out a little bit, thinking about things that are not happening just right at the bottom, but things that are happening in the water column of continental shelves and the edges of continental shelves, we can see some interesting effects on productivity where the nutrient-rich water of the continental shelf meets the less nutrient-rich but more stably stratified water of the open ocean. So the limiting factor for primary production in this well-mixed nutrient-rich coastal water is probably not nutrients. There's a lot of nutrients that are being brought in by the runoff from land and stirred up from the bottom through autochthonous nutrient regeneration. So there's a lot of nutrients here. Nutrients probably not the limiting factor for production, but this water is probably murky because not only are a lot of nutrients being brought up, but there is sediment that's being brought in by the rivers and resuspended by the waves. So these waters are kind of murky. So light is likely to be the limiting factor here. Another reason that the light might be the limiting factor in these coastal waters is if the mixing is going too deep down, and that means that the phytoplankton cells are sort of mixing below the critical depth of mixing. Anyways, in the open ocean water, the problem is probably that not that there's a lack of light, but that there's a lack of nutrients to support the productivity. But right where nutrients limiting, right where these water bodies meet, there's sort of what we call a front, where the well-mixed water mass full of nutrients but murky, and the stratified clear water mass that's stable but low in nutrients, right where they combine, they're sort of like the perfect conditions for phytoplankton growth, where you've got enough nutrients but the water column is stable and well lit, and you get a big bloom of this green growth of algae. And this explosion of algae growth will also attract zooplankton and small fishes and seabirds and marine mammals. So oftentimes sort of around the edge of the continental shelves, where you get the continental shelf water meeting the ocean water, is where you have really, this is supposed to be a whale, really hot spots for lots of charismatic marine species that will come and hang out in these areas because of the food sources there at the fronts. So we're now talking about the roles of light and depth in continental shelf productivity. So we already talked about the role of nutrient availability, and we talked about the role of water currents and waves, and now we're talking about how depth itself and its interactions with light affect the productivity of continental shelves. So as we learned in the primary production lecture, there's a zone of the ocean called the euphotic zone, which is the zone where there's enough light to support photosynthesis. And it goes down, how deep it extends sort of depends on how clear the water is. But eventually you get to a point called the compensation depth, where photosynthesis is no longer enough to make up for the respiratory needs, and that's at about 1% of the surface light. So in the continental shelf environment, the bottom may be shallow enough that bottom is actually in the sunlit zone. And so that means that primary producers can grow on the bottom. And so you get things like seagrasses, kelps, and red algae that can grow right on the bottom instead of just having the phytoplankton that are in the water column. So we call these benthic primary producers, and they include seaweeds, microscopic algae that are on the bottom, seagrasses, zooxanthellae, which are the symbiotic algae that live inside corals. Those are also benthic primary producers. And all those things can only exist if the light is getting down to the bottom. And if you're in an open ocean environment, the light is not getting down to the bottom. The bottom is way down deep here somewhere, and light is fading out to nothing before it gets to the bottom. And so in the open ocean environment, you do have pelagic primary production, but you don't have any benthic primary production. Whereas in the continental shelf environment, you have both pelagic primary production and benthic primary production. So you've got these two types of primary production. And scientists who study primary production and overall ecosystem health in continental shelves, they'll often quantify the ratio of benthic to pelagic primary production. So basically, you measure how much production of seaweeds and seagrasses and kelps and stuff there is, and then you measure how much, which gives you your benthic primary production, and then you measure how much production of phytoplankton there is, and that gives you your pelagic primary production, and you do a ratio. And so maybe if there's a lot more benthic primary production to pelagic primary production, you get like a 2-to-1 ratio. If it's the other way around, maybe you get a 1-to-2 ratio. There's actually more phytoplankton production than benthic production. And this ratio can indicate the health of the environment. So one of the questions that I ask the class when I'm giving this lecture live is, what kind of ratio of benthic to pelagic primary production would be good and desirable? What would you want, and how would that indicate the health of the environment? So what I tell them is that you want a high ratio of benthic to pelagic primary production, and that would be what would be indicative of a healthy continental shelf environment. See, you want the water to be relatively clear and for light to reach the bottom, because benthic primary producers, seaweeds, seagrasses, and so on, they provide a lot of values other than just providing food for the food chain. They provide habitat, and they stabilize the bottom. They do all kind of great things that are better than just providing food. The thing is, if the water is murky because of an excessive amount of phytoplankton productivity, there's not enough light getting to the bottom for very much or any of those plants to grow on the bottom. So in a polluted ecosystem where phytoplankton is at an extreme level and the water's sort of murky green, then these plants on the bottom end up dying, and it skews the ratio so that it's predominantly pelagic primary production with very little benthic primary production. So like a clear water ecosystem with lots of plants on the bottom would have a high benthic to pelagic production ratio, and a murky water ecosystem polluted with excess nutrients would have a low benthic to pelagic primary production ratio, and that is how it indicates the health of the environment. So depth matters for primary production, as I just mentioned. Basically, you can't have plants growing on the bottom if the bottom is too deep and it's not getting any light. But the depth also affects the amount of secondary production that can happen on the bottom, secondary production being the production of animal life. One of the ways that things are connected between what's going on in the water column up here and what's going on in the bottom down here is, well, the term for sort of the connections between what's happening on the bottom and what's happening on the surface is benthic pelagic coupling. Any kind of link in the ecosystem between what's happening in the water column and what's happening on the bottom is called benthic pelagic production. Benthic pelagic coupling. So if there's a flux of organic material or nutrients between the benthic and pelagic environment, and there's generally more flux between the benthic and pelagic environments when the water is shallow. Why is that? Well, it's simply because there's not as great a distance to travel. So it's easier for something from the surface to get to the bottom if the bottom is close by. It's easier for something from the bottom to get stirred back up to the surface in shallow water than in deep water. So it's like a fancy word, benthic pelagic coupling, but it refers to a relatively simple idea that there's more interaction between water column and bottom processes in shallow environments. So let's try to figure out what's going on in this diagram here with all of the arrows. So one of the things that you see here is nutrients in the water column. So this refers to dissolved chemicals like nitrogen and phosphorus that are in the water column. And those nutrients that are in the water column can stimulate water column primary productivity, the growth of phytoplankton, right? So that's not benthic pelagic coupling, that's just pelagic coupling, right? Nutrients in the pelagic environment stimulate phytoplankton in the pelagic environment. But nutrients in the pelagic environment can also reach down and stimulate the growth of seaweeds like this red, green, or brown algae on the bottom. So that would be benthic pelagic coupling. There's sort of a flux of nutrients from the surface down to the seaweeds on the bottom, causing the seaweeds to grow as they absorb nutrients from the water column. So that would be benthic pelagic coupling. Then another way that there can be benthic pelagic coupling is the nutrients might not be directly transferred to organisms on the bottom, but nutrients in the water stimulate the growth of phytoplankton in the water, but then the phytoplankton in the water are eaten by filter feeders. So these clams here are filter feeders. They eat phytoplankton. So the organic matter, including all the nutrients in it, filter feeders, is absorbed by the filter feeders. And then the filter feeders, all these clams like you and I go to the bathroom and release nutrients. And so the nutrients end up in the sediment pore waters, but those nutrients that are getting peed and pooped down into the mud by the clams can then get stirred back up into the water, and then that can sort of re-stimulate more primary production in the water, or could re-enter the cycle here. So you can see there's all kind of interesting transfers of both dissolved nutrients and particulate organic matter between the water column and the bottom. And those kinds of transfers of nutrients or organic matter are benthic pelagic coupling. Here's a specific example of benthic pelagic coupling. It's with filter feeding bivalves in seagrass beds. Bivalves is a word for mollusks that have two parts to their shell with a hinge. So something like a clam or a mussel would be a bivalve. In this case, the bivalve is this shelled organism here. So bivalves consume phytoplankton. And so basically they pump water into through the siphon into their body. They filter out these phytoplankton cells, and so the phytoplankton is a pelagic organism, and the organic matter from the phytoplankton is being transferred into the clams, and then the clams are excreting nutrient-rich wastes. So that's nitrogen and phosphorus, which end up fertilizing the roots of the seagrasses, and that sort of stimulates the growth of the seagrass. So you had basically this pelagic primary producer, the phytoplankton, but it got eaten, and its nutrients ended up getting transferred to this benthic primary producer, the seagrasses. So you're sort of increasing the benthic primary production and reducing the pelagic. And so, as you might imagine, filter feeders like these clams could be pretty important to maintaining productivity of the seagrasses and the health of seagrass beds. What I might ask you to think about is what might cause a flux the other way. So we've sort of transferred organic matter from the pelagic environment into the benthic environment by turning what was once phytoplankton into nutrients and then into seagrass through this interaction chain. What's a way that the seagrass organic matter could then stimulate pelagic primary productivity? Well, one possibility is that something like a sea turtle could come and eat the uh, seagrass. And when the sea turtle then, this is the world's worst sea turtle, when the sea turtle then went to the bathroom, it would fertilize the phytoplankton. So the nutrients that were benthic in the form of the seagrass could then be re-injected into the water column where they would stimulate the phytoplankton. So the theme here is that the transfers can go both ways, like from the water column to the bottom, or from the bottom to the water column, and that those transfers are often mediated by animals. So for the phytoplankton to seagrass transfer, the clams are the mediator, and for the seagrass to phytoplankton transfer, the sea turtle is the mediator. So let's give another example where there are organisms that are mediating a transfer between the benthic and the pelagic environments. The organisms that we'll focus on are these callian acid shrimp, which they sometimes call ghost shrimp. They're these soft-bodied burrowing shrimp that make deep burrows within muddy sediments, and gray whales, this sort of interesting type of baleen whale that feeds not on plankton in the water column like most whales do, but like most baleen whales do, but feeds actually on benthic stuff. So ghost shrimps, the callian acid shrimps, this is supposed to be the surface of the sediment here. And this cutaway view shows that they build these really elaborate networks of burrows, and they live beneath the sediment pretty much all the time. That's why their bodies are sort of pale and poorly defended. And as water circulates into and out of their burrows, it brings oxygen into the sediments, and the organic matter that's in the sediments can sort of decompose, and you end up with this sort of growth of bacteria and fungus along the sides of the burrows of the shrimp. And the shrimp sort of goes along its network of tubes and scrapes away the bacteria and fungus that are growing in the well-oxygenated walls of the tube, and it eats that. And so basically it's turning the detritus in the sediment into secondary production. So it's turning it into shrimp. And these things, their system of having these elaborate burrows in the mud, it works really well, and they can have really high secondary productivity. So basically you generate a lot of shrimp meat beneath the sediment. And this is not lost on the gray whales. The gray whales are able to scoop up these soft, muddy sediments in their mouths, filter out the mud, and extract the bodies of the shrimp from the sediments and eat that. The gray whales, it's kind of a sad story about them. They used to live in both the Atlantic Ocean and the Pacific Ocean, but they were hunted to extinction in the Atlantic Ocean. Now they are only surviving in the Pacific Ocean, with the exception of the occasional stray that crosses through the Arctic Ocean and ends up in the Atlantic. It's possible that with global warming and there being less ice in the Arctic Ocean, that gray whales will actually be able to colonize the Atlantic Ocean again, but we'll have to be careful that they don't die from other things in the Atlantic Ocean, like from getting tangled in fishing gear or poisoned by pollution or the other things that tend to kill whales in the Atlantic Ocean. So the gray whales are sort of a weird branch of the whale evolutionary tree. They're one of the most primitive baleen whales. So baleen is that stuff in some whales' mouths that's sort of like a comb that combs out the um, phytoplankton, usually from the water. Um, and in gray whales, they uh, they have this are thought to be sort of what the ancestral earliest baleen whales were like, where they weren't necessarily filtering plankton straight out of the water, but they were scooping up sediment and using their baleen to filter out the shrimp from the sediment. And they sort of have to twist on their side to do that, and they take a side gulp of the soft mud when they're feeding like this. And so they do this in shallow waters with soft sediments, oftentimes in bays and estuaries along the Pacific coast where they're now found. Okay, we're shifting gears now to talk about the role of bottom type in the productivity and diversity of continental shelves. So the bottom, another word for the bottom is substrate. So substrate type turns out to be really important for determining not just how productive the life can be on the bottom, but what types of life and how diverse the life can be. There's a general trend that hard substrate habitats, like where there's rock or coral on the bottom, tend to have higher diversity than soft substrate habitats where it's just sand or mud. And you can definitely see a lot of productivity and diversity in this picture from a deep rocky reef off the coast of San Francisco. So this isn't your traditional coral reef. The substrate here is sort of different in geological origin. But it is covered with sessile invertebrates, so in that way it's similar to a coral reef, and it's frequented by fishes of all types. So if you want to classify the type of bottom, one of the ways that you can do it is by how big the grains of sediment are. So at the very small end, you've got mud and silt, and then with larger grains, that would be sand, larger still, gravel, even bigger than that, cobble. So you've probably heard of cobblestones. If you think of like one of those old-fashioned streets in a medieval town where they were paved with cobblestones, stones maybe 10 centimeters in diameter. And then boulders or bedrocks would be these large, immovable, or practically immovable stones. So where there's big rocks or bedrock exposed on the bottom, you would call that a hard bottom. And where there's smaller particles, you'd call that a soft bottom. So one of the other things that differs as you go along this spectrum of increasing particle and pore size is, well, the pores. So there's more spaces in between when you're dealing with bigger grains than there are when you're dealing with smaller grains. So that could potentially be a habitat for organisms. Then again, there's some good things about the fine grains, like for an organism that's trying to burrow in, it's easier to push aside the small grains and burrow through sand or mud than it is one of these other kinds of materials. Also, large chunks are more likely to stay in place as waves and currents pass by, so those boulder bedrock habitats might be more stable. Because they've got larger spaces in between them, larger animals might be able to hide in those in-between spaces than in the other habitat. And we'll talk a little bit more about why the habitats with these larger grains might have higher diversity. One thing that I want to say about substrate type is that so the type of bottom influences the type of organisms that can live there, but the type of organisms that live there also influence the type of bottom. So biology affects the substrate type. One example of that would be like if oysters live there, the oysters create these reefs, and then the reefs become a substrate in themselves. Same thing with corals. They basically create hard substrate. So the biology can influence the substrate type, and the waves and currents can also influence the substrate type. If there are strong currents, they might whisk away the fine silt and mud and leave behind coarser sediments and rocks, creating a hard substrate. or if it's an area with still waters, it might allow a lot of fine sediments to be deposited, and you'd have a muddy habitat. And then geology, like the uplift of the land, and if there's volcanic activity making seamounts and things like that, also influences the substrate type. And so all of these factors are kind of interacting with each other, and so it's not like just one is the driver and the other is the response. You have to think of the biological activity is something that is influenced by the substrate type, but something that also influences the substrate type in turn. And you can think of all the interesting interconnections between all of these factors. And so if you're trying to get these concepts straight in your head, maybe look at each of these arrows and try to think of an example in your mind, like, okay, how could biology influence the topography or geology? How could the geology influence the waves and currents? And see if you can think of an example for each direction of each of these arrows. All right, let's talk about organisms that live in hard bottom habitats, so bedrock, boulders, and cobble. They're really good habitats because of their stability for sessile organisms to attach to. So if you've got like a really stable rock, then an organism can stick to that rock and have a firm attachment point even if strong currents are passing by. Whereas if it was trying to grow on soft mud or sand, it might get tumbled away because it wouldn't be able to attach to anything well there. So hard bottom, stable attachment for sessile non-moving organisms. Also, if there's big chunks of bottom in a sea bottom habitat, that means that there's big spaces between the chunks, and those nooks and crannies are large and well oxygenated and good hiding places for relatively large hiding organisms like this Homerus americanus, the American lobster. There are other types of fish that might like to live between the chunks in the rock, like a moray eel. It's not the world's greatest moray eel, but give it some fangs. Angry eyebrows. So holes and hiding places between the chunks of substrate in hard shelf habitats provides a sort of a unique and valuable habitat for a lot of cool organisms, and that's one of the things that leads to the high diversity of these habitats. Another thing that leads to the high diversity of hard shelf habitats is the fact that there's different microenvironments on different parts of the structure. So if you think of a boulder in the water, you've got sort of like a sunny side of the boulder and a shady side of the boulder, and they can be two totally different habitats. The sunny side dominated by photosynthetic organisms like seaweeds, and the shady side covered in sessile invertebrates that don't need sunlight but that filter feed. And then in turn, the seaweeds might have different organisms living in and among them than the sessile invertebrates like the sponges and corals might have. So in addition to providing all kind of interesting hiding places, it provides this habitat variety that can increase the diversity of life in a hard substrate habitat. Okay, well, what about soft substrate habitats? They aren't maybe like quite as interesting to look at as a rocky reef, but they still support a lot of life. And the organisms that do well in these places are crawlers and fast burrowers that can deal with the problems of shifting sediments that I mentioned on previous slides, like getting buried or exposed by changes in the sediment as it's swept around by the waves, having to deal with clogged filter feeding apparatuses as sediments are swept around in the currents. So the organisms that live in these environments are able to deal with that. Organisms that we would call emergent sessile fauna, something like a sea anemone, they're not going to do so well in this kind of habitat because they can't really attach very well, and they'll get swept away. If there is going to be something that's like stuck in this sediment, it would have to be kind of like rooted in. And there are some sessile organisms, like some tube worms, that root into the sediment and they can kind of attach themselves that way and give themselves some anchorage. But even for them, it's hard to attach in an area where the sediments are really shifting around a lot. So you'd find most of these what we call emergent sessile fauna, which means organisms that don't move and that stick up above the sediment surface, where there's less disturbance. Where might there be less disturbance in a soft shelf habitat? Well, maybe, since we know that wave action is severe near the surface, it would be a highly disturbed habitat in the shallow water. But deeper down, you might be sort of beneath the level where the waves stir up. So deeper down, you might be able to have some more delicate emergent sessile organisms that can live down there without getting stirred up by the waves, because the wave action is mainly high above them. So deeper down, you could have some emergent sessile benthos organisms like hydroids, bryozoans, soft corals, and sponges might be able to survive on the soft sediment as long as the currents aren't too strong and there's not waves sweeping them away. So where you do have some bits of hard substrate or larger chunks in the soft sediment habitat, they end up being really important. So biogenic habitat could be something as simple as like let's say there's a clam shell here on top of the sand. So it's just a tiny little bit of hard substrate habitat in the soft substrate environment, but it could be an attachment point for a seaweed or something like that, and maybe a little anemone could grow there. So as soon as you add some hard substrate to one of these soft shelf habitats, you can really increase the diversity. So every little shell or worm tube that sticks up above the sediment adds a little bit of structure to these soft shelf habitats. And so organisms that form structure in these habitats, everything from seaweeds to scallops, can increase the diversity in these habitats. So one organism that can establish itself in soft substrate habitats and then can provide kind of an attachment point for other organisms is this polychaete worm, a segmented worm in the genus Diapetra. So the worm itself is a mobile organism with a mushy body, lots of little palps and antennae and gill-like appendages. But it creates this shell and sand-lined tube that is mostly buried in the sediment. So the sediment would come up to like about here. But the part of the tube that sticks above the sediment, the worm will glue seaweeds to it. And the seaweeds wouldn't really be able to attach to a sand or mud area otherwise. So you end up, it's sort of like you're almost creating a forest where but it's just of seaweeds attached to these worm tubes that are kind of anchored in the sediment. And so you end up with this seaweed bed where all the seaweed is there because it's attached to a worm tube. So you might think like, oh, the worms, they must be seaweed eaters and they sort of attach seaweed to their tubes as sort of their garden, but that's not the case. What they do is they decorate their tubes with seaweeds and stuff because that attracts little organisms like amphipods, little crustaceans, to come and munch on the seaweed. But then the worm lunges out of its tube and eats the little organisms that are visiting its algae garden. So it's sort of a sneaky move by the Diapetra worms. Here you can see a sandy bottom, but you see all of this schmutz sticking up off the bottom. This is actually a bunch of Diapetra worm tubes that the worms have stuck algae and seagrass bits to. So instead of having just a bare mud habitat, you have this kind of structured bottom habitat that provides a lot more structural diversity and opportunity for little organisms to live. And some of those little organisms might become prey for this benthic predatory fish, the lingcod Ophidion elongatus. This is a fish that lives on the Pacific coast of North America. Okay, so we've talked about the reasons why continental shelves are so productive and diverse now. We talked about the role of nutrients, we talked about waves and currents, we talked about substrate type, the difference between soft and hard bottom habitats and the organisms that live on them. Now let's think more about the variety of life that lives in continental shelf environments and how we can sort of classify and make sense of these sea creatures that live in association with the bottom. So we talked when we were talking about plankton, about different ways that we can classify plankton, and now we're going to do some of the similar classification work, but with sea bottom organisms of the continental shelf. So there are at least 6 different ways that you can classify continental shelf organisms. You can classify them by the part of the environment they live in, by the way they move or not, by their size, by their trophic mode, which is their feeding mode, by their taxonomy, which is sort of like where they are on the evolutionary tree, and by a combination of the above methods. So we'll oftentimes end up with compound word descriptions to describe organisms. So these here, they look kind of like dishwashing gloves, but they're supposed to be sea anemones. And sea anemones are epibenthic, which means that they live on the surface of the bottom, sessile, which means that they don't move, macrofaunal, which means that they're pretty big, you can see them with your naked eye, suspension feeding, which refers to their trophic mode, the way they get food particles from the water that drift into their tentacles. Cnidarians, and cnidarians refers to where they are on the evolutionary tree. So here you can see that we used epibenthic to describe the part of the environment that they live in, sessile to describe their mobility, macrofaunal to describe their size, suspension feeding to describe their trophic mode, and cnidarians to describe their taxonomy, and then we combined all of these into this mouthful of a description here. But this is really useful, and I want you to know how to do it. Sometimes pictures help with these classifications. So I've got 2 panels to this slide here. The left panel shows the parts of the environment that we might classify continental shelf organisms according to. So if you are in the water column, you would be a pelagic organism. If you're on or in the sediments, you would be a benthic organism. But there are subdivisions within those environments as well, like the very surface of the ocean, that would be you'd be neustic, that's the neuston. Surface of the pelagic environment you'd call epipelagic. If you live in the water column but near the bottom and in association with the bottom, you're called demersal. So a fish like a grouper that lives near the bottom and in association with the bottom would be considered a demersal fish. There's a grouper with its big lips. And then a fish that lives higher in the water column, like a tuna, would be a pelagic fish. Or epipelagic fish. And then epibenthic means things that live on the surface of the bottom, and infaunal would be things that live in the bottom. And then we can classify them by how they move. So in the right-hand panel, we've got kind of a spectrum from, on the left-hand side, it's things that can move, and on the right-hand side, we've got things that don't move at all, that are immobile. So there's sort of like 3 ways that you can move. You can be a swimmer, and therefore you're considered natant. You can be a crawler, and you're considered reptant. I think the word reptile comes from some Latin word that probably meant crawler. And so reptant means organisms that crawl on the bottom, like this crab. Or you could be a burrower, like a worm. So 3 ways of moving. All these 3 organisms here are mobile organisms, but they're mobile in different ways. And then there are organisms that don't move. So planktonic organisms, they just drift at the mercy of the currents. And so it's debatable whether you'd call that mobile or not, but they don't swim very effectively, so you wouldn't consider them to be natant. Um, they're not rooted in place, so you wouldn't really consider them to be sessile either. But anyways, that's what plankton are. They're drifters. And then sessile organisms, they don't move. They're planted in place. In between mobile and sessile, right in the middle, if we trace this line down, we have these what we call sedentary organisms. So these would be things like clams that they can move a bit, like they might be able to dig themselves up and down in the sand a bit, but they're not going to go on like a long march or swim. They're, for the most part, sessile. They can just move a little bit, so they would be considered sedentary, sort of in between mobile and immobile. Let's talk about classifying things by size. So there's this term macrofauna that's used for benthic organisms, but and so you'd think like, oh yeah, macrofauna, that must be some big organisms. But really it just means anything big enough to be retained on a 0.5 millimeter sieve mesh when you're processing samples. A lot of times benthic ecologists, those ecologists who focus on sea bottom life, collect a clump of sediment or a clump of algae, and they filter the organisms in it through a series of sieves from large to small size. There's various reasons that they might do this. We talked about in the secondary production lecture how sorting organisms by their sieve size can let us use allometric methods for figuring out their biomass and productivity. So that's one reason that we do the sieve size. Another reason is it just helps organize them and make it easier to find and identify the organisms. Anyway, macrofauna are organisms larger than 0.5 millimeter in size, which would be retained on 0.5 millimeter sieve size mesh. Organisms that are small enough to pass through a 0.5 millimeter sieve, but big enough to be retained on a 0.063 millimeter sieve, are called myofauna. I think that means like medium-sized animals. And there's some myofauna shown here. So myofauna are generally multicellular organisms, but they are very small, and because they're small, they're well adapted to living in the interstitial spaces between sand and mud grains. So they're a major component of the animal life in soft sediment habitats, and they're tiny critters. But they're not as tiny as microbiota. So microbiota are like true microorganisms, less than 0.063 millimeters, and those are mostly like single-celled organisms, protists and bacteria. An example of macrofauna would be an amphipod. Amphipods are 14-legged crustaceans, distant relatives of crabs and lobsters, and they're sort of like the insects of the sea. They're extremely abundant in benthic habitats. They're an important link in the food chain. Many of them are plant eaters, some of them are detritus eaters, and they're just like candy for little fish and larger crustaceans that would eat them. So size is important for life, and one of the reasons it's important is that small organisms grow and reproduce more rapidly than large organisms. We talked about this in the secondary production lecture when we were talking about maybe it was the primary production lecture too, when we talked about the production to biomass ratio. So relative to their size, small organisms grow and reproduce faster. So one of the consequences of this is that small organisms like myofauna, they respond quickly. Their populations can quickly increase if conditions become good for them, and their populations quickly decrease if conditions are bad for them. And so if you want to know like what the current status of an environment is right now, you might sample its myofauna, because they respond rapidly to the current conditions, they could indicate recent environmental impacts. So if there was some sort of like a stressor event indicated by this jagged lightning bolt that affected the sea bottom, the myofauna would be quickly affected, and you would see that as a change in the myofauna community. So the little critters that are living down in the sediment would be affected. Macrofauna communities, those made up of larger organisms, they might respond more slowly to disturbance. They might resist the initial disturbance more, but they could take longer to recover from a disturbance because they're larger organisms. It takes them longer to grow, and so that would be things like the clams in the sediment. If there was some sort of event where there was no oxygen in the sea bottom environment, then the clams would die, and it might take months or a year or more for the clams to come back to that environment to recolonize it. And so if you saw in your sea bottom samples there were no clams, that might indicate that sometime within the lifespan of a clam there was some harsh disturbance to that environment. And you wouldn't know that if you only looked at the myofauna, because the myofauna would be able to sort of recover relatively quickly after the disturbance. So that's one of the reasons that benthic ecologists like to look at both what's going on with the very small organisms and what's going on with the larger organisms, because each tells you something a little bit different about the health and the history of that sea bottom environment and when it was last disturbed. Let's talk about classifying things by what they eat or how they get their food. So the fundamental classification here is between autotroph and heterotroph. Autotrophs make their own food. So we've learned a lot about photosynthesis and photosynthetic primary production. Chemosynthesis is also a thing. There's a variety of types of bacteria that can use various energy-rich chemicals as their way of getting power to make organic matter from inorganic materials. And we'll learn more about those chemosynthetic microorganisms when we talk about deep sea ecology. But I won't say anything more about the chemosynthetic autotrophs right now. But I will say something about the photosynthetic autotrophs. So you can see I've used, well, PowerPoint makes this easy to do like bullet point lists where you've got like the higher level and then like the lower level below that and then the lower level below that. And so this indicates the sub-classifications within each group here. So within the autotrophs, you've got chemosynthetic and photosynthetic. Within the photosynthetic autotrophs, you've got various groups like phytoplankton, which we know are planktonic, single-celled photosynthetic autotrophs. Benthic microalgae, which are single-celled algae that live on the bottom. And benthic macrophytes. So that would be multicellular plant-like animals that, or not plant-like animals, multicellular plant-like organisms that live on the bottom, including true plants like seagrass and algae like macroalgae. So this is kind of a sloppy drawing here, but this could probably be some green macroalgae. And these are supposed to represent, these shapes here are supposed to represent inorganic chemicals like ammonia, and carbon dioxide, phosphate, that can be worked into water, that can be worked into organic matter using the power of the sun by photosynthetic autotrophs. Okay, now let's talk about heterotrophs. Heterotrophs should be interesting to you because you are a heterotroph. There are two broad classes of heterotrophs, decomposers and consumers. Both decomposers and consumers, all heterotrophs, they don't make organic matter from nothing. They have to consume or decompose organic matter, and then they rework it into the organic matter of their bodies. That's what heterotrophs do. The energy that they get is also stored in the chemical bonds of the organic matter that they consume or decompose, and that's how they get their energy. So what's the difference between a decomposer and a consumer? This is something that I've been a little confused about sometimes, and I think one of the main differences is that a consumer engulfs particles of food, whereas a decomposer, it sort of leaches out chemicals that cause the food to be broken down into small molecular scale components that can then just be absorbed into the shell, into the cell. So it sort of digests the food outside the body and then absorbs the nutrients into itself. And that's sort of what's the difference is between consuming and decomposing, if you want to get technical. All right, so decomposers, those would be things like fungi and lots of types of bacteria, yeasts, etc. Um, and so consumers, those are animals generally. So there's different types of consumer. You could have predators and scavengers, grazers, suspension feeders, which filter feeders are a subset of suspension feeders, and deposit feeders. So on the next couple slides, I think I'm going to talk more about the consumers and give some more detailed examples of different types of consumers. Right, there's different ways to eat, the different modes of consumption. So suspension feeding is where you're feeding on particles that are suspended in the water. So water, because it's a dense medium, things can float or drift in the water, and there can be a lot of bits of food suspended in the water, and suspension feeders capture that suspended food to eat. So some suspension feeders just kind of wait for the food to drift into them, and they're more passive suspension feeders, whereas other suspension feeders actually pump water through their filter, and those are called filter feeders. So filter feeders are suspension feeders that actively pump water to filter, whereas passive suspension feeders just kind of wait for things to drift into their tentacles. Deposit feeding. So deposit feeding is kind of similar to suspension feeding in that it's feeding on like small particles of food, except that in deposit feeding, in suspension feeding, the particles are suspended in the water. In deposit feeding, those particles have already settled down to the bottom and they're on the bottom. So deposit feeders don't get the particles out of the water, they get the particles off the bottom. And those deposits of food on the bottom are sometimes called benthic detritus. Okay, moving on to the right-hand column here, predation and scavenging. So that's where you're hunting animals. That would be predation, and scavenging would be you're hunting dead things to prey on, carcasses. So we'll talk about how there can be sort of a fuzzy line between predation and scavenging. Grazing is where you're consuming something that's alive, but it can't run away. So like a caterpillar eating a plant would be a grazer, a deer eating a plant would be a grazer, parrotfish eating seagrass would be a grazer. Sometimes the thing that's being grazed is not like one organism, but it would be like a film of algae or that could be being grazed. And then there's parasitism. So that's when the thing that's eating the other thing actually lives inside that other thing. Okay. There are some crossovers and blurry lines between these different categories, but I'll leave it to you to try to wrap your head around those complexities. As an example of the blurry line between scavengers and predators, I'm showing a blue crab, Calanectes sapidus. This organism will prey on live organisms like bivalves beneath the sediment, but it will also scavenge dead materials, like if there's a dead fish or something, it'll be happy to prey on that dead material as well. Demonstrating that blurry line. And so many organisms, even if they specialize in taking dead prey, if there was a living organism that they could easily take down, they might take that opportunity to prey on the living organism. Deposit feeders. So these are the organisms that eat benthic detritus or schmutz that has already settled onto or into the sediment. So many of these organisms crawl on the surface of the sediment, or they dig into the sediment. The heart urchin is a type of sea urchin, a type of echinoderm, distant relative of starfish, that will burrow into the mud. It can completely bury itself. Giant sea cucumber is one of a number of holothurians, another group of echinoderms that consumes large amounts of fine sediments along with the organic matter that's in that sediment, and it will digest the digestible components and poop clean sand out the other end. Grazing is something that we probably think of a sheep or a cow when we think of a grazer. But grazing occurs anytime that the consumer is eating away at a plant, sessile organism, or consolidated layer of schmutz, or what I would call a biofilm. Well, scientists call it a biofilm. I call it a layer of schmutz. Um, and if it's an organism that is eating like a fine microbial film, then sometimes it's hard to, which you would call a micrograzer because it's grazing on a film of microorganism. It can be kind of difficult to separate micrograzers from deposit feeders because deposit feeders are also eating organic matter and microorganisms within sedimentary deposits. So sometimes the line between micrograzer and deposit feeder is a little blurry. The gastropods, like snails and shellless snails, which we would call sea slugs, are a real common type of grazer in marine environments. And there are some of those that graze on plants, like the salt marsh periwinkles shown in the upper right-hand picture. And then there are also gastropod grazers that consume sessile invertebrates. So they're actually eating animals, but they're eating them in a grazing style because they're eating sessile animals, which are plant-like in the fact that they don't run away. So grazing is interesting because of the evolutionary relationship between the grazers and the things that they graze on. The organisms that get grazed on, since they are sessile animals or plants that can't run away, they've evolved a lot of defenses against grazers, toxins or mechanical defenses like spines or things that make them hard to eat or digest, stinging cells. There are a whole array of host defenses against grazers. And the grazers in turn have evolved ways to overcome those defenses, to process those toxins or to be immune to those stingers. And so the evolutionary arms race between the things that are getting grazed on and the things that are grazing on them gets intense, and it results in a phenomenon called host specificity, where there may be host organisms that are so well defended only a few organisms can eat them, and those organisms are so specialized that they can't eat anything else except one or a few species of prey. An example would be the flamingo tongue snail, which grazes on soft corals like sea fans. This is a close-up picture of the flamingo tongue snail. It has a mantle, like the fleshy part of its body, that wraps most of the way around its shell. So the colorful part that you see here is actually the flesh of the snail, and the little bit of white poking through here is the white shell underneath. And as you can see that as this thing trundles across the surface of the sea fan, it's consuming the polyps and leaving just a trail of bare coral skeleton behind. So parasitism is a really interesting topic in both terrestrial and marine ecology that there could be a whole course dedicated to. We're not going to cover it in great depth in this course, but I do want you to know at least some of the classes of parasites in the marine environment. So parasites in general, they're organisms that feed off the flesh and or body fluids of the host. So they could do that from the outside of the host. So ectoparasites, ecto means outside, they live on the surface of the host. So, something like a leech, or there's a type of crustacean called fish lice that lives on the outside of the fish. So here's a fish and it's got like a little fish louse on the outside of it. That would be an ectoparasite. And then an endoparasite would live inside the fish, so that would be like worms in its gut. It's a little cutaway view to the gut, and you can see the endoparasites in the fish. A particularly weird type of parasite is what they call parasitic castrators. These are parasites that don't merely feed off the fish, they sort of trick or the host. They trick the host's sexual system with hormones in get it to behave in ways or change its biology in ways that help it produce more parasites. So one of the most famous types of parasitic castrators is a species of barnacle that instead of being a filter feeder like most barnacles, it's a parasite which burrows into the flesh of crabs and then causes the crabs, even if they're male crabs, to think that they're female crabs and to help raise the eggs of the barnacle parasite instead of raising their own young. Suspension feeders are ubiquitous in marine environments because there are so many suspended particles of food. It's a really convenient way to feed. You basically just open your feeding apparatus and catch the particles coming past. So I've divided up suspension feeders according to mobility. So sessile, which is one of the most common types of suspension feeders, sedentary, and mobile. So sessile suspended feeders need something stable to attach to. That could either be a hard surface or they could have some way of sort of rooting themselves in mud or sand. And the longer and older those sessile organisms are hoping to live, the more stable a substrate they need. Like the hard corals of a coral reef would be very long-lived, very old sessile organisms, sessile suspension feeders, and they need a very stable substrate to stay in the same spot undisturbed for the sometimes hundreds of years that they might live. Sedentary suspension feeders don't move very much, but they do move a little bit. So those would be things like clams that can dig a bit in the sand, but mostly stay in the same place. And clams, of course, most are adapted to living in a soft substrate, but there are sedentary suspension feeders that are adapted to living in a hard substrate. And unlike sessile things living in a hard substrate, they can move and adjust their position a bit. They just don't do it very much. And finally, there's mobile suspension feeders that can swim or crawl. Here's an example of well, it's hard to say if it's totally sessile or just sedentary, but it's a suspension feeder. It lives beneath the mud, and you can kind of see in the diagram here the layer of the mud on the surface. A common name for this is parchment tube worm because it makes this tube for itself that is sort of like rough paper, which they like parchment, this old-fashioned kind of paper. So it makes a papery tube that they call a parchment tube. And there's unidirectional water flow through the tube. So this polychaete worm pumps water with its paddle-like appendages and sort of creates this flow of water that goes out one way and in the other way. And so all of the suspended particles in the water, which could include plankton or other digestible bits of organic matter, get sucked in. And the worm has this sort of mucus net thing that it creates, weaves a net out of mucus, and the particles get caught in that net. Once the net is full, the worm eats the net, particles and all, and then it makes another mucus net and starts doing it again. These things can be really abundant, and you'd never quite know what was beneath the sediment just by the little soda straws that you see poking up above the surface. But these are all leading into U-shaped burrows with one of these strange worms in them. And if you follow the YouTube link, you can see an interesting video where some scientists have sliced the worm out of its tube and put it into a transparent plastic tube, so you can see it moving and doing its thing in sort of a simulated tube. So this is one of the ways that we've studied them and learned about what they do. These are some examples of mobile suspension feeders, ones that swim around as they filter particles out of the water. Anchovies and menhaden, two types of fish that have specially adapted gill rakers to capture plankton from the water. So they swim around with their mouths gaping open, consuming small particles, and they can do this really efficiently to such an extent that large schools of these fishes are actually important in keeping plankton levels from getting too high in areas where these fishes are abundant. And in addition to these small mobile filter feeders, there are large ones like the baleen whales, basking sharks, and whale sharks that I've talked about previously. There are some interesting crabs that with their fierce claws, you'd think that they were predators of some sort that, actively took down large prey. But they, in fact, uh, this species, the porcelain crab, is a suspension feeder. So it just crawls to a good spot to perch. And then with these elaborate mouth parts covered in hairs, it beats these mouth parts around to filter particles of food out of the water. And if that's how it lives, why do you think it has these big claws? If it's just a peaceful filter feeder, why would it even need claws at all? Well, it may have something to do with territorial disputes, to find the right spot and fight with other crabs to maintain its feeding position. It might need those claws. It might also need them for defense from its predators, or perhaps in some sort of sexual selection. The claws would come in handy. So sedentary suspension feeders. So these are sedentary are the ones that don't move much, but they're not totally sessile. So um, some bivalves are sedentary but not totally sessile. They can dig themselves into the mud. This is important in areas where the sediments shift around a lot and you might need to adjust your position as the sediment erodes away or more sediment's deposited. Another reason that moving up and down in the mud can be helpful is that it allows these clams to escape their predators by digging deeper into the mud. And this includes human predators. If you've ever tried to dig for razor clams, you have to dig really fast and deep in order to outrun the clam, so to speak, and catch it. And these things are adapted to living in a soft substrate. They wouldn't be able to burrow into rock like this. They need soft mud or sand. There are some sedentary suspension feeders that are adapted to living in a hard substrate, so they live on the rock or in crevices in the rock, and they use their tube feet here to sort of cling to the rock, and they mostly stay in one spot, and then they have these branching mouth parts that unfurl and can catch particles of food that stick in the feathery tentacles of the mouth parts. And it's interesting to watch these sessile or sedentary sea cucumbers like this, because they have these branching bush-like mouth parts, but they can furl them back into their mouth, and that's what they do to lick the particles of food off of the mouth parts, and then they'll re-extend it sort of like a fern fiddlehead unfurling back into its elaborate form again, and then they'll take the next tentacle and clean it off and furl it back out again, and they'll continue doing that in a circle around their mouth. It's sort of hypnotizing to watch if you're ever scuba diving and have the opportunity to see one of these things feeding. So now we're at the suspension feeders that are truly sessile, that really don't move from their spot. Barnacle is a perfect example. It's literally cemented to hard substrate, and once it settles there, it can't move for the rest of its life. Sponges are similar. They attach to a hard surface and grow in place, and they filter particles of suspended material from the water. And the filtering mechanisms are really different. They've evolved separately because barnacles and sponges are from such different phyla of life. Barnacles are crustaceans. They're more closely related to crabs and lobsters than they are to sponges. And sponges are a really primitive group of life that has some more similarities to protists than it does to other multicellular animals. But both of these have evolved to be sessile suspension feeders on hard substrates. Where it's a soft substrate bottom like sand or mud, it's more difficult for sessile organisms to survive because the instability of the substrate, but they have evolved some pretty complex ways to anchor themselves into the soft substrate. You can see the shell-lined tube of this feather duster worm helps anchor it into the substrate, And this tube-dwelling anemone has a mucus-lined tube that goes down into the sediment and helps it anchor in a muddy sediment so that it can have a basically sessile lifestyle even without a hard substrate. One of the problems with being sessile is that it's difficult to copulate unless you can move over to be closer to your partner. So the way that most sessile organisms deal with this is that they don't have sexual intercourse. They release their eggs and sperm into the water and hope that the eggs and sperm meet in the water. But that's leaving a lot to chance, and if it was possible to copulate, that might have some advantages. And so barnacles, they do manage to copulate despite not being able to move from where they're stuck to the rock. And the way they do that is by they've evolved to have these really long intermittent organs, which is sort of like a penis, and they fertilize each other by via these long organs. They're hermaphroditic, so each barnacle has both eggs and sperm, and so it can both fertilize and be fertilized. So when you think about barnacles living on a rock, you may have seen a rock that had a lot of barnacles on it, and it was pretty obvious that the barnacles were close to each other and they could probably reach over and copulate with each other. If there's just a low density of barnacles, though, then they might not be able to reach each other, and therefore their reproduction would fail. So this failed reproduction that happens when population is at a low density is a phenomenon known as Ali effects. Ali was a scientist. Sometimes my students confuse the word Ali with the term allele, which is something from genetics, but it means a totally different thing, and it's just an unfortunate coincidence that the spelling is kind of similar. Yeah, so the sort of the reduced reproductive rate that experienced at low population densities due to failed mating or other mechanisms is an example of an Ali effect. So Ali effects have a technical meaning in terms of population growth, and so I want to review a particular model of population growth that's important to understanding Ali effects. And that model of population growth is logistic population growth. It's the kind of population growth that if you look at a graph of the number of individuals over time, you see that it increases kind of exponentially for a while, but then it levels off at a maximum population size, which is the carrying capacity K. So I set up the terms in this equation here with R equals 0.01 and K equals 1,000, meaning that the carrying capacity of the environment, the maximum population size of whatever organism is simulating here, would be 1,000. And so as the population gets near 1,000, the population growth rate slows down to 0, and the population is no longer growing once you've reached 1,000. The terms in the equation here, just to review, d and dt equals the rate of change of the population. R is the intrinsic population growth rate, or sort of like the maximum population growth rate per capita. N is the population size. So when you multiply the population growth rate, which is the proportional thing, by the population size, you would get like the actual change in numbers of organisms over time. And then K is the carrying capacity. This term of the equation here is what causes the leveling out, because as the population size n gets close to k, then you have k divided by k, which is 1, and 1 minus 1 is 0, and so basically the population growth rate becomes 0 when n gets near k. So I showed you the population size over time, starting from a slow population and increasing to the maximum size population. That's what we're seeing in the upper left-hand graph here. But I have two other graphs as well, which show the dynamics of this kind of population growth rate in a different way. And the axes on these graphs are different, and it's very important that you understand all three of these graphs and what each of them is showing about logistic population growth. So take the time to replay the slide if you need to review. So in this graph here, the x-axis is different now. It's not time, it's population size. And population size is going from 0 to 1,000. 1,000 in this case is K, the carrying capacity. And then what's on the y-axis is the population growth rate, number of individuals added per unit time. So when the population is very small, like close to zero, the population growth rate is close to zero as well, because the population growth rate is proportional to the population size. And so you can't have a fast-growing population if there isn't anything to build off of, right? So that's why population growth is zero when population size is zero. At the other end of the spectrum, when the population size, and this is also reflected in the sort of the gentle slope of the graph when the population size is low here. And then at the opposite end of the spectrum, when population size is at the carrying capacity K, you also have zero population growth. And that's reflected in the flat slope of the line here as it's approaching K. Where you have a large value for population growth is when population is about halfway to the carrying capacity. Sometimes this is expressed as K over 2. And at that point, that's sort of like where the population is growing at its fastest, and that's where the slope of the increase would be fastest in the other graph. So at this point, you have maximum population growth because you've got enough individuals for the growth to be based off of, but you don't yet have so many individuals that the population growth rate is slowing down and nearing carrying capacity. So it's the point of maximum population growth. Interestingly, maximum population growth rate occurs when you're only about halfway to the maximum population size. So in the graph in the lower right, we have the same x-axis. It's still population size from 0 to K. But what we're showing on the y-axis is a little bit different now. So now instead of showing total population growth rate, which is R times N, we're showing the per capita population growth rate. So that's the population growth rate per individual. So it's sort of like each individual's contribution to population growth. So that is a maximum when the population size is low and a minimum when you reach carrying capacity. So this is the thing that's a little bit tricky to figure out. But imagine you're like bacteria. So here's a bacteria. It's just a simple rod-like cell. And if you're the one bacteria in a big petri dish, you've got that whole dish to yourself. You've got all of the resources in that petri dish. You can divide and create more bacteria and divide again and create more bacteria. And so your population growth rate, if you're this one starter bacteria, is going to be really, really high when you're like the first one colonizing this area. And that's represented by when the population size is low, like you're the first one to colonize the new area, your population growth rate per capita, per individual, is really high. As the petri dish gets crowded with more and more bacteria, the population growth rate is increasing, but per individual bacteria, their output is decreasing because it's a little bit crowded for them. They're not able to produce as many offspring because their offspring are having to fight for resources with the other bacteria in the dish, until you get to the point where you can't possibly fit any more bacteria in the dish. You've reached the carrying capacity, or all the resources have been used up, and you could have a bacteria in there, but it wouldn't be able to reproduce at all, and its per capita population growth rate would be 0. So basically the downward slope of this line, decreasing per capita population growth rate from when it's just one organism in a virgin environment with all the resources to exploit, and of course its population is able to expand rapidly from that initial colonist all the way up to this point. So you'll notice that I used a bacterium as my example for this organism that has maximum population growth rate when it's just like one introduced to a virgin environment. For organisms that reproduce sexually, they might not have their maximum population growth rate when the population is a population of one. In fact, you wouldn't be able to reproduce very successfully if it was just you on a desert island. You need at least one other person to make any population growth progress. And that's where allay effects come in. So not all species experience allay effects, but for the ones that do, you have this graph, which you'll notice this one looks very identical to the graph that I showed on the lower right of the previous slide. So you've got this per capita population growth rate that declines from low populations to high populations until it's basically zero when you reach K. If the population experiences allay effects, though, and remember, allay effects are the reduction in per capita population growth rate at low population sizes, then you have reduced population growth rate not only when you're nearing K, but also when you're sort of below like a critical threshold of population density, then population growth rate starts to decrease per capita there, and that's sort of indicative of lower reproductive success for whatever reason when you have low population density. So you probably would not see an allele effect for some asexually reproducing bacterium, which would be able to reproduce just great when it was the only one in the dish. But for something where mating is important, then allele effects might be more likely. So to summarize, in a population with allele effects, there's less reproductive success per individual at low population sizes because of the allele effects. But for all populations, whether they experience allele effects or not, you have reduced reproductive success at high population densities because of the carrying capacity thing. So all populations experience reduced population growth rate at the high end because of the carrying capacity issue, which is true for all organisms, but only some organisms, depending on their reproductive mode and other aspects of their biology, also experience reduced population growth rates at low population densities, and those that do, we call it allele effects. We're shifting gears here from this, kind of esoteric aspect of population growth ecology to more of the ecosystem scale description of marine continental shelf environments now. And this is what I'm going to finish the lecture with, talking about some of the variety in continental shelf ecosystem types around the world. So not all continental shelf environments have the same kind of productivity characteristics. Where this has come up with earlier was when we were talking about primary productivity, and we talked about the spring bloom and secondary fall bloom of phytoplankton as you go from winter to spring to summer to fall to winter again. And so some ecosystems have a certain type of seasonal cycle and overall dynamics, and other ecosystems don't have that. They would have something different. And this scientist Longhurst, who's a biogeographer, remember biogeography means life earth mapping, where you're sort of mapping the patterns and trends of life across the earth and how it varies from place to place. So Longhurst, in addition to describing the biogeographic provinces of the ocean, described different continental shelf ecosystem types. So types of marine continental shelf environments that had certain characteristics that he could describe. And so I'll go through and I'll talk about each of the 7 types of continental shelf ecosystem that Longhurst described. So the first type of continental shelf ecosystem that Longhurst characterized was a type that's found only at very high latitudes where it's permanently covered in sea ice. So the primary producers in these ecosystems are under ice algae. So you can see how the lower part of the ice surface here looks green because it's stained with algae. And these under ice algae don't do much in the wintertime because it's dark 24 hours a day in these areas in the wintertime, but they become fairly productive in the summertime. And the ice, though it doesn't totally melt, it might partially melt, and these under ice algae can also sink to the bottom. And although it's not shown in this picture, you'll see in the video that on the bottom in these ecosystems there are often really, really diverse, well, maybe not super diverse, but really productive ecosystems with lots of interesting invertebrates that feed on the algae that filters down off the ice or grows on the bottom. So these ecosystems have sort of like a brief period each year of enhanced productivity when the light's shining through the ice, but otherwise have sort of low productivity during the dark months of the year. As we move a little lower in latitude, we get ecosystems that are covered with sea ice for part of the year, but not the entire year. And in these kind of ecosystems, there is phytoplankton bloom when the ice retreats. There's some stratification in the water based on salinity as the melting ice sort of freshens the water and creates this sort of salinity-based stratification in the water. So there's like a lower salinity in the surface layer and higher salinity in the deeper layer. And this kind of stabilizes the water column and allows a phytoplankton bloom to happen in the water as the ice is melting. But because the growing season is so short in these areas, their seasonal cycle of phytoplankton is just kind of like one peak in the summertime, and that's it. So it can be really productive, but it's just like sort of one bloom per year during the time that the area is ice-free. Then we've got this classic spring bloom, fall bloom system in the continental shelves of areas at mid-latitudes, temperate zones. Not all temperate zones have this kind of system. Usually it's temperate zones with wide continental shelves and minimal upwelling, where the mixing in the wintertime and destratification is sort of the main way that the nutrients are brought up to the surface. So you have sort of this wintertime where you've got a lot of nutrients but no big algal productivity because of a lack of sufficient light, and then you get the spring bloom, but the season is long enough that the spring bloom depletes the nutrient levels, and so the bloom declines, but then there's a secondary bloom in the fall. And I won't describe this in too much more detail because I've already described this in the primary production lecture, but this kind of seasonal cycle happens in Longhurst's type 3 continental shelf ecosystems. The Atlantic Ocean from Canada to Florida would be an example of a place where we get these kind of bloom cycles. Then there are continental shelf ecosystems in temperate waters that don't have this spring-fall bloom cycle, and they call them topography forced continental shelf ecosystem types. And the continental shelves, these really productive waters off of Alaska, are an example of this type of continental shelf ecosystem. And they've got wind and tidal mixing that interact with sort of the shape of the coastline in the bottom here. And it means that there's sort of like a constant mixing and or upwelling that keeps nutrients available all year. So you don't get this boom bust kind of productivity. You just have kind of a general increase in productivity during the sunnier part of the year and somewhat of a decrease during the darker part of the year. In certain parts of the coastlines of the world, particularly on the west coast of continents, and particularly where the continental shelf is narrow, so there's sort of like a quick drop off to the deep sea, you get these upwelling driven continental shelf ecosystem types, where there are prevailing wind patterns that cause nutrient-rich waters from the deep sea to deliver nutrients to the continental shelf and stimulate strong primary production during the times that upwelling is strong. And the upwelling is usually strongest in the summertime because that's when these systems have their strong winds that move the surface waters offshore and bring the deep cold water up from the bottom to stimulate the primary productivity in these upwelling systems. California, sort of Oregon to California, is the classic upwelling ecosystem of the United States. In the tropics, there are two, Longhurst divided them up into two kinds of continental shelf ecosystems. There's the river runoff dominated tropical system, tropical continental shelf ecosystem type, where it's warm year round, plenty of sunlight, but it's murky, and there's not a lot of coral reefs because there's too much nutrients being brought in from the rainy runoff and rivers gushing into the ocean. So you can kind of see the phytoplankton blooms along the coast and swirling through here, and the water looks kind of murky and green. It wouldn't be a great place for scuba diving or snorkeling, but there could be a lot of fish productivity and other marine life productivity in this area because of the abundance of nutrients. And then the final continental shelf ecosystem type is tropical, but it's not heavily influenced by river runoff. And so these areas are low in nutrients, they have clear water, and this allows this particular ecosystem type, the tropical reef ecosystem type, can exist there. And because you've got this growth of corals which produce a lot of calcium carbonate rock, you have a lot of carbonate sand that's sort of contributes to the sediments in these areas, and structures the ecosystem with this limestone rock created by the coral, and then the carbonate sand created as that rock wears down, and organisms here that are adapted to being very efficient in a environment that has clear water and lots of sunlight but is low in nutrients. And that's all that I have for this continental shelf lecture.